Uh, Good day, wherever you find yourself. And Chris, thanks for the introduction and for the invitation. Today, I give a talk on deciphering the genetic etiology of syndromic orofacial cleft by integrating both our Sub-Saharan African dataset and also resources in FaceBase. As you said, I'm currently a faculty member at Kwame Nkrumah University of Science and Technology, abbreviated as KNUST in Ghana. And that's our university, the area view of our university, and that is our entrance, and that is our demonstration. Uh, okay, so a bit about the orofacial cleft. As we know, this condition, it's originate when the facial prominences fail to fuse as we expect during embryology, normally from the 4th up to the 12th week. We expect both the lip and the palate to form in humans. Any perturbation as far as the fusion of this process are concerned, and then as well as the palatal shells result in clefting. We know this has multifactorial etiology, especially so with the non-syndromic forms. And with the syndromic forms, depending on the phenotype we are looking at, we believe that about 50% of cleft palate, for example, are likely to be syndromic, whereas when it comes to cleft lip and cleft and palate, probably about 35%, 30% of that are likely to be syndromic. Now, in Omnium, there are over 500 syndromes, for example, that may present with clefting. And that is very important to us, especially in our setting where genetic testing is rare, for which reason it is difficult for us to know exactly the syndromes underlying the various cleft presentations that we see in our setting. This image is from our previous study showing the various phenotypes, for example, the midline clefting, that is macrostomia, for example, that is bilateral clefting. And apart from that, there are a few instances we also see tarsal cleft as well in our setting as well. And in our clinic, for example, most cases are actually sporadic. There are no familiar history. And so, as I said, syndromic clefts are very important. Worldwide, we know that Van der Waals syndrome are the most common cause of syndromic cleft, accounting for about 2% of all this. And this is a case of Van der Waals syndrome from our center, for example. And we know that if you pick that syndrome alone, about 70% of them may be due to mutations in IRF6 gene. For about 30% of them, we may not find IRF6 gene mutations. We know now, for example, about 5% of them that present with cleft palate may have mutation in the green hair-like 3 gene. And quite recently, our group shows that there are instances where we can also find mutation in PRCK1 gene in individual that lack mutation in RF6 gene. But still, for example, if you pick the syndrome, there is some misinheritability. That is to say, there are certain cases that we are not sure we can't find mutation in any of these genes. Of course, people have said that apart from these exonic mutations, there could also be mutations in regulatory sequences like promoters, enhancers, that could also influence this disease. But still, there is some misinheritability that we are yet to find as far as this condition, for example, is concerned. So in essence, even with this, what we see as a single syndrome, there may be multiple genes contributing to it. And that is expected because typically in living in genetic systems, hardly will any gene work in isolation, just they work in a pathway, for which reason when members of the pathway get mutated, we are likely to see very convergent phenotypes. So for example, in our center, one of the syndrome that is easy to diagnose will be the Van der Waals syndrome. So for our study since 2012, we've been collecting data. So for those that are obvious, of course, when it's Van der Waals syndrome, we can just pick it. and then do sequencing. And indeed, we've done quite a number of them, and all of them have confirmed RS mutations in them. All right. So interestingly, for our cohort that we've designated as clinical as Van der Waals syndrome, interesting for us, all of them we've been able to detect RS mutation in them. So it's been easy for us to detect the genetic etiology of Van der Waals syndrome in our cohort by just doing targeted genome sequencing of the RS gene. And these are a publication, for example, my days as a graduate student in Iowa, and these are other publications from our group. Now, so put into perspective, in our clinic, for example, because there's no genetic testing, for example, the multidisciplinary group come together, and then we look at the cases, and then, for example, determine whether they are syndromic or non-syndromic. So, for example, based on this paper by syndromic group, by clinical evaluation, looking at the presentation at the time the patients show up, we estimated about 12% of them are syndromic. And obviously, it's obvious at the age of manifestation, that is the age at which we do the clinical evaluation. Of course, there are many of them that the phenotypes are not common, are not easy to pick up at the time that we do the clinical evaluation. So, for example, we follow the group by 3 months, the lip is repaired. By 12 months, 9 months to 12 months, the palate is repaired. And unfortunately, once the repair is done, hardly would this patient return to the clinic for follow-up. So it's easy to follow them up and to pick up the subsequent phenotype that may show up. So it's always easy, it's difficult to actually always do the syndrome diagnosis based on just the clinical phenotype, especially so when they present very early to the clinic and they hardly show up once the repair is done. So that is obviously one major thing that is limited syndrome diagnosis. All right. So if you pick our center, for example, there are, apart from Van der Waals syndrome, worldwide we say the most common, but if you pick our center, for example, Pierre-Robin sequence is very common among us, more common than Van der Waals syndrome. So we see more cases of Pierre-Robin sequence. And for example, cleft presenting with liver abnormality, cleft palate, these are more common than the Van der Waals syndrome in our cohort, for example. But the issue that we know that PRS, for example, is not a single syndrome. Stickler syndrome, and even in cases of Treacher-Collins syndrome may present with Pierre-Robin sequence characterized by the micronathia, glossoptosis, and sometimes cleft palate, for example. So it's difficult always to look at what syndrome we are dealing with, especially so when genetic testing is not common in us. Of course, we've done many other, for example, using GWAS data, we've been able to determine some trisomies in our system, in our setting. We able to look at deletions, structure variations, and all those ones. But again, these are usually done during research because genetic testing is not part of our setting here. So this is, for example, a group, a cohort presented with clefting and then clapfoot, for example, and then we show that multiple syndromes may actually present with that. So that suggests to us that when we see that any presentation, for example, clefting and then limb abnormalities like clapfoot, we may not be dealing with a single syndrome, with multiple syndrome. But unfortunately, as I said, because of this, at the time of examination, we are able to pick up all the phenotypes. So that has been the limitation of that. So as I said, ideally in typical birth defect clinic, we expect that genetic testing should be part of the clinical care. But unfortunately, the infrastructure here is not well built. Not many trained personnel. We don't have any policy on incorporating genetic testing into our healthcare system. And obviously, we know how the cost of genetic testing. So in our clinic and elsewhere within the country, genetic testing is on the routine part. For few individuals who could afford it, whenever genetic syndrome is suspected, we have to do this outside the country. And that is the challenge that we have in our system. So obviously, in our cohort, apart from, as I said, Van der Waals syndrome, that is easy to be so certain about a few other syndrome. we don't do any genetics testing to confirm this kind of syndrome. So again, the trouble has been that we know that, as I said, a lot of the syndromic cases, we hardly do the repair. That is to say, uh, hardly do they survive to the time of where the repair is done. And we know that even in literature that the survival rate of syndromic cases are lower. And it's reported, for example, among in our certain infanticide could be very high whenever there are such defect. And all these are many reasons why it's difficult to manage such conditions. So our study is based on this background where the data set that I shared with FaceBase is based on this background where we are not sure of the many syndromes that we are dealing with. So we had to do exploratory study, picking up some of the syndromic cases. Of course, that exclude the obvious Van der Waals syndrome, and then the Down syndromes, and then of course the structure, the gross chromosomal abnormalities. But there are many others that we are not sure what we are dealing with. So of course, we had to document the phenotypes very well so that we can share that. But then until we do genetic tests, we are not sure of what we are doing. So the aim of this study was to do a preliminary study, pick up about 40 of such families, and to undertake whole-exome sequencing. And that will help us to decipher coding variant that may underline the etiology of this, and also look at secondary findings. So in exome sequencing, that is our genome, entire DNA. We are interested in the regions that actually encode the proteins. So we are going to fragment our DNA and then pick up the regions that encode the proteins. And then those are the regions that we are going to sequence to determine the sequences, and we are comparing it to the reference genome. And that tells us which regions are not shared between our individual with a condition and those without condition. The study design is to get sample from the affected individual and then the unaffected parent as well, so that the parent will serve as internal control to help us look at which variant may be causing the disease. So that has been the study design. So as I said, case parent trios, the challenge with obtaining trios, for example, in our setting, is usually when the clefting occurs. A lot of the fathers, unfortunately, abandon their families. And for that matter, it's easy to get mothers than obtaining a sample from fathers, for example. So that has been the challenge. So WES is very important. Whole-exome sequencing is a very good approach at a clinic because it's cheaper compared to whole-exome sequencing. There's less data to manage. The exome is just about 1 to 2% of the genome, entire genome. So it's easy to manage it. And again, it's also very important because it can help us to pick up a lot of coding variants from different populations. Again, we know that about 85% of pathogenic variant actually occur within the coding sequence. So it's always a good an avenue to start looking for variant that may be causing genetic syndrome, especially when you are dealing with syndrome that you suspect they are Mendelian in nature. Okay. So the data set we generated are made up of 120 individuals from 47 families. So 26 of these families are case parent trios and the 21 are case mother dyads because we struggle to get the paternal samples. For some of them, you have to travel to wherever the families are staying, but unfortunately, you are not able to get the fathers to participate. So that has been a challenge generating trio data set for such individuals. All right. Again, only probands. So our families are simplest families where the proband is the only affected individuals. All right. So again, the sequencing was done using the Illumina HiSeq. It was performed at read length of 100 eggs, for example, and then it's a paired end read. So we decided to do relatively deep sequencing so that increases the accuracy of the variant calls. And again, we also use the paired end read as well. And this also helps us, for example, when we are dealing with copy number variation calling, that also helps us as well. So the data sets being shared is available in FastQ files. And we are going to also share VCF files that were, for which the variant calling was done using the Sentinel pipeline. Of course, once you have the FastQ files, we can use the GATK, Sentinel, whatever pipeline you want to generate the variant VCF files. So about ethics, about the data sharing, this data set is approved for sharing. The RRB was obtained from both our teaching hospital called the Confanachi Teaching Hospital and our university, KNUST. There is a Committee of Human Research, Publication and Ethics, also approved. And this is just a screenshot of the consent language in our consent form. For example, telling the participants that we are likely to share this data and even the sample. For example, by sending them to, of course, when I was a graduate student, that's where my mentor Jeff and then Aziz introduced me to FaceBase. So since then, we've been sharing sample and then data with the FaceBase repository. So it's in our language that this data set, the samples may be shared with FaceBase, dbGaP, and many other ones, and the reuse of the samples, and then the reuse of the data set and everything. So that has been in our consent language to make sure that the data is shareable or reusable by others as well. So the data set could be accessed via Facebook, FaceBase, and this accession number. And it will be under controlled access because of the sensitivity associated. That's the kind of consent the participant give because of stigma associated with birth defect in our setting. And again, so we need to protect the patient privacy. Right, so that will be there. Again, the data will not be currently available, or there's embargo on it because we want to, again, publish from it before the embargo is lifted. I'm sure within the next 1 year, it should be available to the public. And also the issue is that genetic research is quite rare in our setting. So our university promotes that, requires that in sharing the data, we also seek collaboration as well so that we have the opportunity to also to be trained about it. Our university also maintains an active FAA number to make sure that our research is done in an ethical manner. So again, the data that we are sharing is follows the FAIR principle. So as I said, we can find it, the data set in FaceBase, FastQ file formats are available so that you can use any bioinformatic tool for the variant calling. And we're also going to share the VCF files that we generated through the Sentinel pipeline, for example. Of course, the demographic and clinical data is also available as well. That will accompany the genomic data set so that you know the kind of patient you are dealing with. Again, the data is also accessible using any standard pipe, as I said. Whatever variant calling pipeline you want to use, once you have the FastQ files, this from an Illumina platform, you should be able to use those for that purposes. And then interoperable, as I said, whatever workflow that you want to use, once you are going by community standards, you're able to arrive at the same VCFs that we are using over here. And as I said, our ethics promote the reusability of the data through the consent and RRB approvals. And then we also, the ownership of the data, where we collected and everything is all available to make it easy to do, to access it. So this is just borrowed from the concept of fair data, and then our data meets that. Now, why is Sub-Saharan African data sets very important? For example, I picked this from the GWAL diversity panel, for example. As it stands, if you look at the contribution of African participants, just less than 1% to GWAS. But if we look at the discovery phase, the participant and the association coming up. So for example, if you pick it, the participants we said is about 0.55%. But the association coming up as much as 10% is coming from African data set. And this is not surprising considering how diverse the cohort is and So in essence, though few Africans have participated in GWAS as far as the discovery phase is concerned, they've contributed significant to as much as about 10% association. In essence, probably using African data sets has a great potential of revealing more associated loci, for example, in this instance. And of course, as diverse as this population is, this is very important. So that is a Ghanaian map. And we are located somewhere here. So that's where our data center is collected, is situated. But unfortunately, interesting, we have people coming from all around the country because this is where we have our National Cleft Care Center. So that's where it's located, and that's where we do our collection. And again, this is back to the GWAS diversity panel, looking at the contribution of GWAS, and purely we see that there is a lot of work in North America, in Europe, but obviously Africa, it's highly underrepresented. But we know that without population-specific data set, we are bound to misrepresent data information. So, for example, variant that may be classified as pathogenic elsewhere, or even among Africans, may not be pathogenic per se, just because they are underrepresented in the population databases, for example. So it's always important that we generate African data set to do that. But unfortunately, the investment by our local government have not been toward that direction. So we are not doing a lot of sequencing. For example, all the sequencing that I'm doing are sponsored largely by NIH and then also by IDR, for example. All right. So without this African specific data set, how are we going to promote the precision medicine that we are promoting? That will be difficult to achieve when we don't have population specific data set to do that. So that's the essence of that. Again, it's very important that we get African data set because of, for example, differences in how or clinical presentation or the facial profile of people. Of course, we are dealing with craniofacial phenotype. And there have been many studies showing that, for example, Africa may have relatively broader face than the European face, for example, for which reason there are facial variations. So probably if you are not familiar with a particular facial variation, you may misconstrue as normal, but probably that may be a disease phenotype, for example. So there were, for example, days where people thought that probably Down syndrome did not exist among Africans. And it was because of probably the facial profile of Africans, so it was very difficult for us to pick up. So that's Down syndrome in different individual, for example, and that's a typical African. So if we are not very conversant with that, we may not see this as Down syndrome, for example. And for example, there have been studies in, for example, presentation 20q deletion syndrome as well, where we see the facial difference between Africans and other populations look different. So it's always very important to get clinical phenotypes or images, facial images from different populations so that we're able to make very good meaning out of the phenotypes as far as the genomic sequence from different populations. Unfortunately, as I said, this data set are largely lacking. So these are few manuscripts that are under review from the data set that we are sharing. That is a manuscript on cleft presented with limb abnormalities. We also have one on Pierre Robin sequence. And the beautiful aspect about, as I said, we are presenting new syndromes and probably we are showing new variant and probably new candidate genes that we associated with it. And as I said, for example, because of the deep sequence and the paired read, we could use the data set, for example, for copy number variation calling as well, and using 4 different tools and a consensus approach, for example, we're able to detect the SHH variant and many other copy number variant in this cohort, for example, will also be contribute to Clefton. So again, the challenge with that, ideally in our in filtering variant, after getting the variant, or the gene, you must be able to make meaning out of it. And ideally, we should be doing functional studies. But unfortunately, as I said, normally we do have limited funding over here. So that we have to fall on many resources like FaceBase and others to make sure that we're able to make meaning of that. So whenever I have after our filtering or prioritization, for example, using the CMG guideline, we have to come back to FaceBase, look at what this gene has been reported in FaceBase, what may be the expression pattern of the gene, what are the facial phenotype that have been associated with the disease. So we always come back here to make sure that we can make functional meaning or add additional information to this. So these are some of the resources that we've been using in FaceBase to make sure that we're able to make functional meaning of our, as I said, how do we determine the functional craniofacial relief of the genes that we detected in humans? So we have to fall on FaceBase, look at how the gene reported, what are the phenotypes, what is the functional relevance of those are very important to us, the expression patterns, and if there are other omics data set on that gene, so that we make in silico functional meaning out of the various data set that we are generating. So these are a few references. And then I acknowledge my NIDCR, Fogarty, and IADR for supporting this research. And thanks to my mentors and collaborators for helping with the work we are doing here. So thank you very much for having me.